Assalamualaikum warahmatullahi wabarakatuh and good afternoon to Dr. Nurul Huda and all my fellow friends. My name is Muhammad Arrahman bin Jamil, BR21110082. I would like to present about the impact of elevated carbon dioxide concentration on the nutritional properties of Solanum lycopersicum L, which is tomato plants. As we all know, the global ecosystem is at risk due to human activities fueled by population growth and increased consumption of fossil fuels, which have led to rising levels of carbon dioxide. This CO2 has a significant impact on plant growth, increasing the yield and altering key enzymes, which ultimately results in more biomass in C3 plants. However, although these changes are well known, the underlying mechanisms are not fully understood. This raises concerns about potential effects on agricultural productivity, plant quality, and ultimately global food security. Of particular concern is the potential for a high sugar diet from carbon dioxide plants to disrupt nutrient balance in human diets, potentially leading to health issues such as insulin resistance and other metabolic disorders. The objective of the research is to investigate how increased atmospheric carbon dioxide (CO2) levels influence the nutritional composition of Solanum lycopersicum L. tomato plants. The method that have been used for this research: first, plant material and sample preparation. Seeds of tomato variety Solanum lycopersicum. There are three types of seeds, which are Stupici, Plovit, and LA0147, were sterilized to the method described by Eppenroth and germinated as described by Boreja. After germination, the plants were transferred to soil potting and kept in culture chambers under the flowing conditions, temperature of 22 until 23 degrees Celsius, and two different concentrations of CO2, ambient CO2 400 ppm and CO2 900 ppm. The plants were sprinkled with a fertilizer solution NPK. The tomato fruits were harvested at the last stage of ripening, then stored at -80 degrees Celsius. Secondly is mineral content. Samples were collected from the three independent plants and then was digested with nitric acid. On the following day, the samples were incubated in the hot block for 8 hours and 85 degrees Celsius. There are many ingredients and contents in tomato. Firstly, micronutrients, which include carotenoids and lycopene content. Secondly, macronutrients, which are sugar, lipids, and proteins. And thirdly, antioxidant bioactivity. And the last, total polyphenol content. The final results of this research. First graph is for the mineral content. Mineral content of magic tomato fruits were collected from three plants for each accession. Under ambient 400 ppm of elevated 900 ppm atmospheric carbon dioxide were used for analysis. As you can see here, there are three types, and first is LA147, second Clovit, and the last seeds is 2PC type of tomato. Next graph is for micronutrients. Carotenoids content of tomato fruits were collected from plant grown under ambient 400 ppm of elevated 900 ppm atmospheric carbon dioxide. A is for total carotenoid content and B is for lycopene content. As you can see here, At the 400 ppm, the value of total carotenoid content for 400 ppm concentration of CO2 is higher compared to 900 ppm concentration of CO2. For the lycopene content, the 400 ppm is higher than 900 ppm concentration of CO2. Next is for macronutrients, which is sugar content. Carbohydrate, carbohydrate content of magic tomato fruits were collected from plants grown under 400 ppm of elevated 900 ppm atmospheric CO2. A is for sucrose content, B glucose content, and C fructose content. And as we can see here at the graph, for ST 400 ppm and ST 900, ST 400 is higher sucrose compared to ST 900. For PV, Plovit tomato, the PV 900 has higher sucrose compared to 400 PV. For LF tomato, the LF 400 is higher value of sucrose compared to LA 900. The value of sucrose is not stable for each tomato seeds that have been compared. The next graph is for total lipid and protein content. Lipid and protein contents of magic tomato were collected from plants grown under 400 ppm of elevated 900 ppm CO2 concentration. A is for total lipids and B is for protein contents. As you can see here, the 900 ppm has higher lipids compared to the tomato seeds of under ambient 400 ppm CO2. But for the PV, it's not same. The PV 400 is higher compared to PV 900 ppm. I think it is because of the genetic background of Plovit tomato seeds. The next graph is for antioxidant bioactivity. Fruit samples were collected from plant grown under ambient 400 ppm or elevated 900 ppm CO2. A for total polyphenol content, B for 2,2-diphenyl 1-picrihydrazyl, and C for oxygen radical absorbance capacity or SS. For the graph A, total polyphenol content, they are so list of difference between 400 ppm and 900 ppm. But as you can see, as at the Stupici tomato, 400 ppm has higher value of polyphenol content compared to ST 900 ppm. And then we move to the next graph at B. They also not not so many difference between the value of 400 ppm and 900 ppm concentration of CO2. And for the C, we can see here for ST 400 is lower compared to ST 900, and for PV same also, the PV 400 is lower than PV 900, but not for LA. LA 400 has higher value compared to LA 900. I think it is influenced by genetic background. The next graph is nitric oxide production. The effect of Stupici tomato fruit extracts of on nitric oxide liberation and scavenging capacity. Fruit samples were collected from plant grown under ambient 400 ppm or elevated 900 ppm CO2. A is for fruit extract inhibition rates on nitric oxide production by J774 microphage. B graph is fruit extract nitric oxide scavenging capacity. For there are no difference between 400 ppm and 900 ppm because the two graph is decreased. As the discussion of the results, we will into the effects of CO2 on phytochemistry and bioactivity potential in a wide range of magic tomato varieties which represent a significant portion of the genetic diversity found in large fruit types. Tomatoes are known for their abundance of bioactive compounds essential for good health such as minerals, vitamins, carotenoids, and phenolics. By examining the impact of CO2, we have observed noteworthy discrepancies in mineral content, specifically in sodium, potassium, copper, iron, manganese, and calcium levels, suggesting the it, that its influence varies depending on the cultivar. These findings highlight the diverse effects of carbon dioxide on large fruit tomatoes as compared to smaller fruit varieties. Furthermore, the changes in mineral intake induced by CO2 could potentially have implications for one's health by disrupting electrolyte balance, immune system function, and other biological processes. As the conclusion, we can conclude this study delved into the impact of elevated atmospheric carbon dioxide at 900 ppm parts per million on the nutrition and health benefits of magic tomatoes. Surprisingly, levels of sucrose, fructose, and glucose did not increase, while protein, carotenoid, and lycopene contents actually decrease under CO2 conditions. Additionally, antioxidant and anti-inflammatory properties did not show any significant changes. These unexpected results highlight the fact that the effect of elevated CO2 on the nutrition and health potential of tomatoes is influenced by genetic background, challenging commonly held assumptions, and environment. More research is crucial in order to fully comprehend this intricate response, providing valuable insights for decision making in agro-food health systems and aiding the push for healthier food options in the face of climate change and the growing prevalence of metabolic disease. We would like to acknowledge Dominic Pellet, Perry Bratt, Thierry Golly, and Sabine Galindo for their scientific advice and Manon Vito for technical support. As you can see here, this is my journal reference that I have been used for this research and my poster. That's all from me. Thank you.